I'm Greg Lang. I'm here to tell you about yeast experimental evolution. It's long been appreciated that when populations adapt in parallel, they tend to find similar solutions to the same evolutionary challenge. So take cavefish, for example. So cavefish have evolved multiple times independently from the same surface fish. And when they do, they tend to do so in very parallel ways. For instance, they lose pigmentation, they lose eyesight. And this parallelism actually extends to the genetic level. So both of these populations have different mutations in the same gene underlying loss of pigmentation. So how surprising is this? My research really aims to answer the question, how reproducible do we expect evolution to be? And we don't do this using fish, we do this using yeast, and there are many reasons why yeast is a great system for this. For instance, yeast grows very quickly, 10 generations of growth per day. Yeast can be maintained as haploids or diploids. They can be propagated sexually or asexually. And we actually know more about yeast than we do any other eukaryotic system. And so when we find changes that happen in response to the selective pressure, it's easy to understand why those changes may or may not have occurred. So what is experimental evolution? Well, so you can imagine doing the following experiment. You can come in one day and inoculate a bunch of test tubes with yeast, or in our case, we use 96 well plates. You can let these cultures then grow to saturation, so you can let them grow overnight, come in the next day and dilute them all. And you can just repeat this process over and over and over again. And with 10 generations of growth per day, it doesn't take very long to get thousands of generations of evolution for hundreds of populations. And then after some time, we do some sort of analysis. Usually in our lab, this involves doing next generation sequencing. So we don't actually do this using dilutions by hand, is that we have robotics in the lab that we use to aid in this process. And so a student might come in the morning and take the plates out of the incubator, set them up on the deck, press start on the robot, and the robot will do all the dilutions. And a half hour later, you've diluted hundreds of cultures, something that would take you hours if you had to do by hand. And so after some time, we actually start seeing changes in these populations. So what might we see? So here are 3 examples of populations that evolved, and here we're looking through the bottom of a 96 well plate, and you can see that some populations get some weird morphology. So in the middle one, you can see a selective sweep of some really strange looking pellet morphology. And in other cases, not much is visible. Maybe the pellets get a little bit bigger, as you would expect if they're adapting to their environment. But when we really see things is when we do whole genome sequencing. And so we can do sequencing on these populations, and we can do time course sequencing, sequence the entire population at multiple time points over several thousand generations. And when we do that, we reveal all sorts of complex dynamics. And so we can see every single mutation that arises in this population and reaches some appreciable frequency. We can see mutations that piggyback on one another. We can see mutations that are competing and out-competing and going extinct. And it gives us a really detailed picture on the dynamics of adaptation. In addition, we can say something about what biological processes are being changed in this environment. So for example, we can take all of the mutations we find and ask if we see the same genes hit multiple times in independent populations. And the answer is yes, we do. So here's a list of the top 24 genes, I think, that we see hit over and over again by mutation in different populations, and they tend to fall in the same biological processes in negative regulators of RAS, things in the mating pathway, and we know that when we do an evolution experiment, we select for sterile strains, much like in the cavefish, you select for loss of pigmentation and loss of eyesight. And we also see things involved in cell wall biosynthesis and cell wall assembly. In addition, we can take all of these 21 mutations that have hit the IRA1 gene, and we look at them, we can see a mix of missense and nonsense and frameshift mutations, which suggests that when we're getting mutations in IRA1, these are loss of function mutations, and we're selecting for loss of function of this negative regulator of RAS. In contrast, however, when we look at the cell wall assembly genes, we only see missense mutations. And this suggests that we're not selecting for loss of function, but we're selecting for alteration or attenuation of function of these genes. And so we get a very detailed picture on how cells are adapting to a particular environment. In addition, yeast are a great system for genetics. And so we can take some evolved strains. So here's one that picked up 5 mutations over some amount of time, and we can simply cross it back to the ancestor, which has none of those mutations. We can then let Mendel shuffle the deck, and we can get all sorts of random progeny from this cross that have random combinations of these evolved mutations. And then by measuring fitness of these segregants, we can say something about which exact mutations have a fitness advantage, what is that fitness advantage, and whether or not there are any genetic interactions between these evolved mutations. For instance, mutation A and B do nothing by themselves, but when you get both mutations in the same background, you get a large benefit. This paradigm of experimental evolution is actually quite versatile. And so we can inoculate haploids or diploids, different strains, different species, hybrid species, and we can do evolution under the same conditions. In addition, we can change how we do the growth. We can change the environment. We can have fluctuating environments. We can ramp the temperature of the incubator up 1 degree every 100 generations and ask if evolution can keep up with that change. We can do it in sexual populations, like I mentioned. In addition, we can change how we, or more appropriately, how the robot does dilutions to change these fundamental population genetic parameters like population size. We can introduce migration between populations. And of course, we have our very powerful downstream analysis of doing fitness assays and doing next generation sequencing. So I hope you enjoyed this very brief description of what we do in the Lang Lab. We hope to see you here. Thank you.